All right, let's go with mRNA processing. Now, this is something that happens in eukaryotes only. For a variety of reasons, prokaryotes do not process. No processing of mRNA in prokaryotes. Okay, only eukaryotes. All right. So after our mRNA is transcribed, it gets processed in order for it to be allowed to leave the nucleus and to get translated properly. Okay. So we modify pre-mRNA before the genetic messages are dispatched to the cytoplasm. So during RNA processing, both ends of the transcript are altered, and sometimes some interior parts of the molecule are cut out and other parts are spliced together. Okay, so let's have a look at this process. So there's each end of the pre-mRNA is modified in a particular way. The 5' end, or kind of the beginning of the molecule, receives what we call a 5' cap. And the 3' end gets a poly A tail. Okay. Now these modifications are for a couple of reasons, and they help to protect the DNA. That's kind of protect the RNA, which is good, from being chewed up and destroyed before it gets translated. They seem to facilitate export of mRNA from the nucleus to the cytoplasm. And they also help ribosomes kind of find and attach to the 5' end. Okay, so let's look at this. So the cap is actually a guanine triphosphate. Okay, and that gets put on the 5' end. And there's a series right here. UTR stands for untranslated region. Okay, and that means it's not translated. Okay, and then there's the start codon that tells us where to start, the segment that encodes the protein. There's a stop codon, which tells the protein needs to stop being made. There's a 3' UTR, or again, untranslated region, but it contains a polyadenylation signal, okay, which is our second modification. Here is a poly A tail, and it's just like poly means lots and A means adenines. So there's many, can be hundreds of adenines long, okay? And this kind of gives us our modified RNA that's then ready to be exported to the cytosol, unless it needs to be spliced, okay? And splicing actually helps to explain a lot, because the human genome has 25,000 genes, which sort of seems like a lot, but when you really think about it, it's not that many for the complexity of our bodies. And it turns out that RNA splicing, we'll talk about this a little bit after I tell you what it is, sort of solves the problem of how it seems like we really don't have very many genes. It kind of comes up with an explanation as how we can kind of make more of what we have. Okay, so anyway, so a lot of eukaryotic genes have these long non-coding stretches of nucleotides that are kind of nestled in between the coding regions. And the coding regions are the ones we're interested in. That stuff codes for protein. The non-coding regions are called intervening sequences, or introns. Oops. There we go. Introns. The coding sequences are called exons because they are expressed. Okay. RNA splicing removes the introns and joins up the exons together, and it creates an mRNA molecule with a continuous coding sequence. So let's see what that looks like. So here we have our pre-RNA, we have our cap, we have our poly A tail, okay? And then here is an exon, and you see this section right, I'm going to change color because it's hard to see that. What should I change? Let's go with the blue again. Okay. Here's an exon. Okay. That does not code for it. Nope, that does code for something. The intron doesn't code for anything. Here's our next exon. This codes for something. This is good. And then there's this other intron. This is just garbage. And then our third exon. So what we basically need to do is, in order to make our protein continuously, we just need exon 1, exon 2, and exon 3 to be put together. So what splicing does is it cuts the introns out, okay, and splices or joins the exons together. Okay, so here, so exon number 1 is here. Let's say exon number 2 is there, and exon number 3 is there. Okay, and then we have a continuous coding segment that will properly translate our protein. And now, because before we had a pre-mRNA, it needed to be modified, it needed to be spliced. And now, once it has its cap and its tail and it's been spliced, it's called a mature mRNA. And that gets exported to the cytosol, okay, finds a ribosome and gets translated. Okay. Now, RNA, the splicing of RNA is carried out by a very exciting thing called a spliceosome. And it's just cool. So a spliceosome is a complex of proteins and several small nuclear ribonucleoproteins, which we call SNRPs. And the SNRPs base pair with the splice sites and recognize the splice sites, so they guide the proteins that are going to cut the RNA to the right spots. So let's see how that works. So here we have my pre-mRNA up here, and we have our transcript. Here's the intron, change my color again, that I'm going to want to cut out. There it is. So I want to cut that out. So we take some SNRPs and some other proteins. These are enzymes and stuff. Okay. And they all get put together into a spliceosome. And as you see, the SNRPs recognize and base pair to the appropriate points in the introns. And they've kind of fold this up and they bring the two ends of the exons together, close together, so that when the enzymes cut the RNA, the appropriate points, it ligates or brings the two exons together, and the intron is removed, and it has this structure here, and it's called a lariat. You may know what a lariat is. Another name for it is a lasso, like the cowboys use to capture little wayward sheep and stuff like that. But this kind of looks like a lasso, or a fancy name lariat, and so that's why we call it a lariat. Okay, so that's how the spliceosome works. I want you to remember this lariat thing for a second, because we're going to talk about that right now. I mentioned this way back when we were talking about proteins and enzymes and things like that, and I said not all proteins are enzymes, and not all enzymes are proteins. I said because there are some enzymes that are made of RNA, and here they are, finally. So ribozymes are RNA molecules that have catalytic activity and can behave like enzymes. And these ribozymes are the lariats. Okay, so a lot of times these lariats that are cut out of the mRNAs, these introns, can serve as ribozymes. So it's kind of cool. Okay, before the discovery of ribozymes, everybody thought that all enzymes were proteins, but that is not true. Most enzymes are proteins, but not all of them. Okay. there are three cool properties of RNA that enable it to function as an enzyme. It can fold up and form three-dimensional shapes because it can base pair with itself. And we kind of saw that back here with the SNRPs. See, the SNRPs kind of fold up and make this funny little shape here. And when we look at tRNA, or we have looked at tRNA, tRNAs fold up into funny shapes as well. Okay, ribosomal rRNA also folds up and becomes part of the ribosome. So it can form three-dimensional shapes. It also, some of the bases in RNA, like adenine, guanine, cytosine, uracil, they contain functional groups that can participate in catalysis. Okay. And RNA may also hydrogen bond with other nucleic acid molecules. It can bond with DNA, it can bond with other RNAs because of the nature of base pairing, because of the rules of base pairing. And so these ribozymes often participate in reactions that require cutting or manipulating of other nucleic acids like DNA and RNA. Okay. Oh, Aspen, cut it out. All right. So evolutionary importance of introns. And this is kind of what I was alluding to earlier. Okay. So first of all, the introns can be used as ribozymes. What's cool is that some introns actually contain sequences that can regulate gene expression. So the intron itself may code for something that controls the expression of other genes. Okay. Some genes can encode more than one polypeptide depending on which exons or which segments are treated as exons during splicing. Okay. So what this is saying, and this is really kind of cool, so listen up. It's saying that one gene can code for more than one protein. It used to always be thought one gene, one protein. But if you splice your introns and exons together different ways, alternative ways, okay, you can make different combinations of coding sequences, okay? And then that can make different proteins, all from the same gene, okay? So that means that the number of proteins an organism can make is much, much, much greater than the number of its genes. So remember before when I was saying, we have 25,000 genes, which when you think about it really doesn't seem like enough. But because of alternative splicing, each gene can code for many proteins. There are some genes that actually code for hundreds of proteins. And those are some of the genes that are involved in your immunity. Okay. So it's really kind of neat. All right. And proteins have this architecture with discrete regions called domains. And a lot of times different exons code for a domain, like exon 1 will code for one domain, exon 2 will code for domain number 2. Okay. So not only can we splice these together differently to form different proteins, but we can also mix up the order of the proteins. And that's called exon shuffling. Okay. And exon shuffling can make all new proteins evolve, which is really kind of neat. So let's look here. If we look at our gene, we have exon 1 codes for domain 1, exon 2 codes for domain 2, exon 3 codes for domain 3. And then depending on which ones we splice together and the order that they're put in, we can change the entire nature of the protein, because if you change the structure of the protein, what do you change? If you change the structure? Oh, that's right. You change the function. And that's how you get all these different proteins. Okay. So that's exciting and very interesting. And that's all I have for now. The end.